This is a walkthrough of the neuromuscular junction, excitation-contraction coupling, and the cross-bridge cycle. An action potential travels down a neuron, and voltage-gated channels open up that allow for calcium cations to enter. That calcium triggers the release of acetylcholine into the synaptic cleft, exocytosis. Waiting on the other side of this cleft are receptors for the acetylcholine. Those receptors, when filled, open ligand-gated channels for sodium and potassium. Sodium enters, potassium exits. They're both +1 cations, but more sodium is entering than potassium, so it has a positive effect on the cell, which is inherently negative. So a depolarization occurs. Yeah, the cell going from mostly negative to more positive. Not necessarily neutral, but more positive. So the depolarization can occur, voltage-gated sodium-potassium pumps to open along the sarcolemma, but it also allows the action potential to travel down into the cell. Action potential travels down the sarcolemma into T tubules, which are invaginations of the sarcolemma. It's really just a continuation that travels down deep into the cell. The plasma membrane of the muscle fiber doesn't just stay on the outside in this specific type of cell. So, travels down the T tubule, which buddies up next to the sarcoplasmic reticulum, which is where calcium is stored. Along the edge of these two structures are voltage-gated calcium channels. When they're activated by the action potential, calcium is released into the cell. Calcium, while out and about, is looking to bind with troponin, which we'll see a close-up here in a second, is along the green here, which represents actin, and the red is myosin. So, calcium is looking to bind with troponin, which are the blue aqua circles, which troponin is actually attached to tropomyosin. So when calcium is bound to troponin, troponin kind of gets pulled back, which pulls tropomyosin back too, and it exposes binding sites on the actin. And myosin, shown here in red, is always looking to bind with actin, always ready and waiting. So as soon as it's available, myosin binds with actin. As it binds, it uses energy originating from ATP, but after it hydrolyzes it, it's ADP and plus that phosphate. So it moves forward, and then, so it's spent, it's on actin, and then ATP shows up, and it's pretty fond of ATP too, so it actually lets go of actin and binds with the ATP. It hydrolyzes the ATP and does this sort of cocks itself back, essentially, into like a ready position until it sees actin. We have to go back here until it sees the actin is available again, because it's no longer ATP, it's only ADP and phosphate, so it's really not as interested, so it binds again. And this continues on and on and on until it doesn't. Until calcium, when it leaves the troponin, the tropomyosin covers those binding sites again, and myosin is just left waiting for myosin, for actin to be available again. So this continues, and as the myosin continues to bind with the actin, it moves it little by little, well, very quickly, but it moves it a bit at a time, moving the actin, moving the two Z ends, I guess, is a better way to put it. Let me go back here. These are the Z lines. Pretend this is one sarcomere here. Oh, that's hard to point to. Okay. These two here. So neither the actin nor the myosin is changing in size, but it's more inching the actin in together to create a contraction. Okay, so then the action potential, it stops on the neuron. Myosin, sad. The action potential stops. So acetylcholine isn't being released. The acetylcholine leaves by way of diffusion, which just happens just naturally over time, or acetylcholine esterase shows up, that breaks it down, and where it leaves not so magically, and back into the exon terminal to once again become acetylcholine down the road. Okay, so without acetylcholine then, the sodium from the muscle fiber, losing my train of thought. Here, 1 second. Okay, so without acetylcholine, everything essentially can't happen. So the sodium then is being pumped out. And without the sodium there, the calcium is being pumped back into the sarcoplasmic reticulum. There's a calcium ATPase pump that puts it back in. And then our myosin and actin, they relax. And the sodium, potassium, the general ratio, the cell becomes repolarized. It goes back to how it was naturally. So let me go back to the more interesting picture. When acetylcholine and all of this doesn't really react, this neuromuscular junction, when it doesn't work like it's supposed to, different diseases can arise. One of them is an autoimmune disease called myasthenia gravis. It's an autoimmune disease, which means that like your body turns on itself. Instead of like antibodies attacking things that shouldn't be in your body, it starts actually attacking itself. And in this case, the antibodies actually attack these receptors here. And without having as many receptors, you have a bunch of acetylcholine just kind of floating around and not actually doing anything. But the stuff that does bind doesn't allow you to move as much, since you don't have as much acetylcholine, you don't have as much movement. So people with myasthenia gravis are typically, they get tired really fast and they're weaker. I mean, sometimes there can even be like a droopiness, I guess, to their face, and it really affects a lot of facial muscles. And that's all I got about the neuromuscular junction, excitation, contraction, coupling, and the cross bridge cycle.